Hi students, today's topic is molecular biology topic, very interesting topic and also important topic, right? You have two essay questions, one is transcription, another one is translation. In these two essay questions, I am going to give the subtitles for these two essay questions. Before that, the repeatedly asking question is post-transcriptional modifications, another one is post-translational modifications, right? You should know the difference between, very carefully read the question, right? Post-translational modifications, right? You know the meaning of translation. After translation, you will get a polypeptide chain which is actually immature in form. After some modifications only, this polypeptide will get converted into mature polypeptide. That is called functional protein. Otherwise, no use of that translated protein. Right? This is the idea of post-translational modifications. Okay, now we will see what are all the modifications. The first one is foremost and important one is proteolytic cleavage. For post-translational modifications, first point you have to mention is insulin. Actually, it is synthesized in the form of proinsulin. Then this proinsulin is converted into insulin. This is called proteolytic cleavage, one of the post-translational modification. Right? Second one is for this conversion of proinsulin to insulin, you have to draw the diagram. Right? First proinsulin diagram with the alpha and beta chain, then insulin diagram, how many amino acids like that, disulfide bonds like that you have to mark. Diagram is very important for this proteolytic cleavage. Second one is activation. Actually, before this, that is after this proteolytic cleavage, we are having some TION, Shun reactions, right? Examples of 5 shuns, that is activation, hydroxylation, carboxylation, phosphorylation, glycosylation, acetylation, methylation, like that. Okay, so I am going to give one by one. Activation means trypsin and chymotrypsin GIT enzyme, you know very well. These are all called zymogens, ZYMOGEN, zymogens, that is inactive precursors, which will be converted into active compounds depending upon the situation, right? The first one is activation, example is trypsin and chymotrypsin. Next one is hydroxylation. This is also very important and you know very well. In ascorbic acid, hydroxylation, that is hydroxyproline and hydroxylation, which is involved in the collagen formation, right, with the help of ascorbic acid. If there is any defect in this ascorbic acid, means they will go for scurvy. For this also you have to write some reactions or diagram. Next one is carboxylation. This is coming under again vitamin, that is fat soluble vitamin K cycle. Here what happens is glutamic acid of prothrombin, that is clotting factor, involved in blood clotting, which undergoes the influence of vitamin K. Vitamin K only converts the carboxylation of glutamic acid, which is involved in blood clotting. So carboxylation. Next, phosphorylation. In carbohydrate metabolism, glycogenolysis, the regulatory enzyme is glycogen phosphorylase. So this is the example for phosphorylation, which is regulatory enzyme for glycogenolysis. Next one is glycosylation. Here the example is immunoglobulins. This glycosylation only will give the immunoglobulins to stability, secretion and cell recognition. These three are the important points for the immunoglobulin glycosylation. Next is acetylation and methylation. Both occurs in histones, that is histones in gene regulation for acetylation, whereas methylation leads to histones in gene expression. The next one is disulfide bond formation. You know the peptide bond, protein bond is disulfide bond formation. Example insulin and also in thyroid hormones. And the last one is, suppose this post-translational modification won't occur properly. What happened? You are having more number of immature polypeptide chains. Definitely, they will produce some diseases like scurvy in vitamin C deficiency, immunoglobulin activity will be lost, and hemoglobin produces anemia, from anemia to hemoglobinopathies like sickle cell anemia, thalassemia, like that. So this is about post translational modification, your university 5-mark short notes. If it is your essay question, definitely you have to add this post translational modifications with the diagram. Okay, bye.